Hello, everybody, and welcome to our second workshop for Ever Wonder How Scientists. Today, we're going to be looking at aquatic invertebrates. These workshops are hosted by us here at the River Institute and specifically by the Great River Rapport project. And they are one of the really important indicators that the River Rapport has been talking about and looking at, and you'll learn more as the reports and the stories come out from them. And we really like to look at invertebrates because they're a great indicator, especially of pollution. So some invertebrates are really, really sensitive to pollution, other invertebrates a little less so, and we can actually look at the different types of invertebrates that we find in an area and use that as an evaluation of what the water quality there might be like. And they're also a really intrinsic part of our food webs. So they're all the way at the bottom of the food webs, and they feed small fish, which feed bigger fish, which later on feed us. So they're also really important to look at our health, as well as the health of the river. So we're going to take you guys out. We're going to go live into the field with some different types of equipment. We'll show you a few different methods that we use for sampling invertebrates, and then we'll bring you back into our lab and you'll get to see some specimens live under the microscope. So I'm going to get geared up. Even though we're not going to be going very deep into the water, it's always really important for us to wear our life jacket because accidents happen. Even in shallow areas of water, it can be unsafe if anything happens. So I'm going to get geared up. And I've got my partner in crime here with me, Steph, and we're going to do some invertebrate sampling. So I will carry this if you want to go to. Yep, I'll get that. And you guys can follow us along. So today we're live from the River Institute, and we're lucky enough to be located right on the St. Lawrence River. So we'll take you down into the little bay here to do some sampling with us. All right, so I'm gonna stand away from stuff here so I can take my mask off. So we're gonna sample, like I said, some aquatic invertebrates. And for those of you who don't know what aquatic invertebrates are, aquatic means relating to water. So that's why we're down here in the little bay. The invertebrates live in the water, and invertebrates are animals that don't have a backbone. It comes from the word for vertebrae, which make up our backbone, our spine. And so the invertebrates we're sampling are mostly going to be in the sediment on the bottom of the river here. So we've got a few different tools that we're going to show you how we might use them. Our most preferred tool is this net right here, which is in the shape of a letter, a backwards letter, but we call this a D net because of the shape of the hoop on it. We also have a kick net, and if you want to pass me one of the poles, you would normally use this net with 3 people, 2 people to hold the poles out like this, and a third person to kick the sediment and the invertebrates into the net. So that's how you would use this one. This one right here is an Ekman Dredge and gets you a pretty even sample of sediment. So the way that this one works is there's these little loops on the bottom, which you would hook up. The other one's broken on the other side, but you would do that on both sides, and it's basically like a big shovel that you would kind of drop down into the sediment, and then you would push the release on the top here, which lets down the 2 sides of the scoop, and when you pull it up, all of the sediment would be inside of this little tube right here. So that's how you'd use an Ekman Dredge, but because the D-net is our preferred method, especially the easiest one to use when you're just one person, Steph's going to show us a little bit how you would use the net, and I can explain as she is demonstrating. So the method we use with this type of net is called the kick and sweep. And like I said, the invertebrates really live down into the sediment. So Steph's gonna dig down with her toes She's gonna do a little twist, a little dance, a little boogie, and sweep the net back and forth in a figure-8 circle through the water. Now she's not dragging the net on the bottom because we don't just want to take big scoops of sediment. We really want to get what we've disturbed up into the water column. And she's going to continuously sweep while she's moving because these invertebrates are really good swimmers and we want to make sure that they can't swim out of the net while we're sampling. And Steph's going to go just to her knees for right now for demonstration purposes, but oftentimes we'll have to sample about 10 meters out into the water and we can end up waist deep, sometimes chest deep, trying to sample these guys. So for today, we think probably 1 scoop is pretty good. At this time of the year, we get a lot of invertebrates, we get a lot of variety, and Steph can show you what it looks like inside of the net there. And a lot of the time when we bring students out into the field and we do this program, they look in their nets and they say, Lexi, I don't see anything in there. I don't see anything moving around. Just kind of looks muddy and gross. There's no invertebrates. I didn't catch any. And I always say to the students, if your net looks like this, if it's full of sand and sticks and algae, I guarantee you that there is some invertebrates in there. So Steph's gonna dump what she's collected into the bucket. We usually put a little bit of fresh, clean water into the bottom of the bucket before we put our invertebrates in there. And then we invert the net into the bucket and give it a good rinse so that everything that's collected inside of the net, all those little bits and pieces are put into the water. And sometimes we have to do this if the sides are really coated, if lots of little bits are stuck up around the top of the net to make sure that everything gets rinsed out properly into the bucket. And I particularly like these nets with the long tails because they're a lot easier to invert into the bucket when you're done with them. Not all of our nets are like that. Some of them are a little bit more shallow, but I like this one. And Steph's got something on her finger there. It might not be super easy for you guys to see on the screen like that, but we'll definitely have more of these guys to look up close under the microscope once we get back into the Institute. So you guys can follow us along as we head back into the Institute. We're going to take this bucket up to Brian, who is in the wet lab, and he is going to show you guys these inverts up close. So we're going to head back up this way. Wait.
All right, so you should be seeing my screen now. They're on the way to bring me what they've just collected, but this is actually our second sample. So my name is Brian Hickey. I'm a research scientist here. I also teach courses on identifying these benthic invertebrates. What was that, Zach? So the first one you're seeing here, it's red. It's called a bloodworm, and I always tell my students, try not to learn some of these common names because common names of invertebrates are really misleading. So it's called a bloodworm, but it's not at all related to a worm. It's actually an insect. This little worm-like animal, I can tell it's not a worm because you can see it's got a little head and a couple of little appendages near the head. If you're lucky, you might even see a little antennae, and that tells me it's not actually a worm. This is an insect. This is a coronamid. Coronamids are midges. Midges, when they're adults, look a lot like mosquitoes, but they don't have sucking mouth parts, so they're not feeding on your blood. They're sometimes annoying you if you're out by the water and you see these tiny insects in giant clouds swarming around your head. You're probably seeing midges. And the reason they're red is most insects don't have anything that resembles our blood, and they do have fluids that transport oxygen, but the fluids are typically not red like blood. These worms have a pigment that's related to hemoglobin. It's very much like the hemoglobin in our blood that helps carry oxygen throughout our body. And this gives this animal an advantage in very low oxygen conditions. They can survive in very polluted, very oxygen poor environments. So if you find only these it's a really bad sign. If you find a few of these, but lots of other organisms that require lots of oxygen, then you're probably dealing with a healthy river or stream. So these, so that's a bloodworm. That's the common name. A better name for it is a Coronamide or a midge. I'll look at our next one here. So if any of you are gardeners, this might look a little bit familiar. These, this is an isopod. Isopods are crustaceans, but they're not insects. They have too many legs to be an insect. You can see the legs here. And if it looks familiar, you might see something a little bit similar to this in your garden, which are often called sow bugs or pill bugs. Another example where common names are misleading because they're not bugs. These are also crustaceans. They're related to things like crayfish and shrimp. And when the ones you find in your gardens, the ones that are called sow bugs or pill bugs, when they're disturbed, they often roll up into a ball. Their defensive behavior is kind of like that of an armadillo. They roll up into a ball and they just present their outer protective shell to all of the would-be predators. So that's an isopod. So there are soil isopods in our gardens. There's also aquatic isopods like this species here that are pretty common in the St. Lawrence River and elsewhere. And another group that is sometimes confused with isopods, whoops, are these guys here. So these are amphipods. So amphipods look a little bit like isopods, but amphipods are flattened from side to side. One of the common names for these guys are side swimmers because when they're in a dish of shallow water, they kind of flop over on their sides and swim around like this. So these side swimmers and these isopods, but especially these amphipods, are really important sources of food for fish. Perch in this area at certain times of the year will just gorge themselves on these amphipods. So they're important. They're fairly high in fat content, so they're good nutrition for perch, especially in the spring when the water's really cold and a lot of the other insects haven't hatched yet. They'll feed on these amphipods. So amphipods, if you have amphipods in your water, it's a pretty good sign. They don't do well in extremely polluted water. But this next one here, this is something we really like to see in our samples because these are Ephemeroptera. These are mayflies. And mayflies are, especially when they're immature, when they're nymphs, we call immature insects either nymphs or larvae. I'm going to use those two words interchangeably. So a nymph or larvae means an immature, a baby insect. So this is a mayfly. It's got those three tails, and that's one of the things that tells me it's a mayfly. And mayflies don't survive unless there's sufficient oxygen in the water. So it's always a good sign when you find mayflies. In fact, one of the really simple ways of assessing water quality is to do a calculation, calculate an index called an Etp Index, which stands for Ephemeroptera, Trichoptera, and Plecoptera. And those names may not mean much to you, but these are stoneflies, mayflies, and caddisflies. And if you have a large number of different species of those three groups, you're probably dealing with some pretty decent quality water. So if you don't know what a mayfly is, mayflies are those insects that often cling to screen doors if you have a property near the water. Their wings stand up over the middle of their back, and they're very triangular, like giant sails on a sailboat. They retain those tails, those tail filaments when they're adults. Sometimes the adults only have 2 tail filaments rather than 3, like the nymph. So mayflies are always good things to have. Another thing we commonly get are these things. Now, it looks a little bit at first glance to a mayfly, similar to a mayfly, because it has those 3 tails. But whereas the mayfly's tails were threads, you can see these look almost feather-like. You can see these filaments here where my cursor is look like feathers. And these are actually the animal's gills. In a mayfly, those tail filaments are just that. They're tail filaments, they're tails. In this one, these feathery tails are actually its breathing apparatus. They are its gills. And this is a damselfly. And so it's an insect. The damselfly is a type of insect. So unlike the isopods and the amphipods, these nymphs only have 6 legs, 3 pairs of legs, which you can see here. You can also see one of the other characteristics that makes something an insect is it's got a distinctive head and thorax and then abdomen. And the wings in an adult insect and the legs in both adult and immature insects always attach to the middle segment. So the thorax starts here and it ends right here. And you can't really see the connection very well because of this big air bubble, but you can actually see a distinctive connection here where the thorax ends and the abdomen begins. So that's a damselfly. When people see adult damselflies, they often confuse them with dragonflies. So this is the damselfly right here. It's very, very tiny. It looks a little bit like a dragonfly. A dragonfly holds its wings out like this, like an airplane shape, whereas a damselfly looks just like a dragonfly, but it holds its wings parallel to its body and just on top of its back here. So that's the difference between a dragonfly and a damselfly. These are indicators that water is kind of medium quality. It's not necessarily perfect water quality, but it's also not extraordinarily polluted. Now this is an immature insect, and scientists call these ceratopogoniids. Ceratopogoniids are another group of midges. So midges are those insects that are a lot like mosquitoes, but I talked about the coronamids, which are midges that are mosquito-like. Just trying to get this better focused here. This guy moves pretty quickly. I'm going to see if I can find one that's less active here in my dish. So these midges here, these are another type of midges. These, the ceratopogoniids, the one that's swimming too quickly for me to keep in your view here. These are the biting midges. So these are those insects that people sometimes call no-see-ums. They're so tiny, you don't see them, they land on you, and you only know that they're there when they take a big chunk. Well, not really a big chunk because they're tiny insects, but it feels like they take a big chunk. There, finally, it's staying still a little bit. I'll see if I can focus a little better. So these are insects in the order Diptera. So these are members of the insect group Diptera that includes all the true flies. Most insects have 4 wings, 2 pairs. Flies, Diptera, have 2 wings. And so this is one of the groups within the Diptera, the 2-winged flies. So other groups of two-winged flies would be houseflies, mosquitoes, the non-biting midges or coronamids I showed you earlier. And these insects here, they can survive a variety of different conditions. They're not necessarily an indicator that water is extremely clean or extremely polluted. Let's see what else I can find here. And so when we're doing this, if I was doing this not as a live stream to show you guys what scientists are up to when they're studying various things in the water, what I would be doing is I would be not only identifying every different type of organism, I'd be tallying up the relative numbers of each of them, and I would be putting it into a formula that's been developed, and there are a number of different formulas that would produce a biotic index, a number that summarizes on a scale of 1 to 10 or 1 to 8. There are different indices and different scales you can use. They all roughly do the same thing. They give you an overall score representing how healthy the water is. And so this, whoops, where'd it go? This one here is an aquatic mite. So many mites are parasites, but there are, and so mites are 8-legged creatures. So that means they're arachnids. It means they're related to spiders, but they're not spiders. They're in a separate subgroup called mites, or in this case, this is a water mite. Scientists call them hydracnids, but water mites are works fine. You can see it has 8 legs. Well, they move the legs pretty fast, so maybe you can't count them. But if we were to preserve this and slow it down, you could easily count 8 legs. And so most mites are parasites, but the ones we find in aquatic sediments and aquatic invertebrate samples are free-living predators. So they're running around quickly the way you see this guy doing, and they're eating smaller organisms, so zooplankton and other smaller invertebrates. And if those of you who are just out sampling, Stephanie and Lexi have found some things that I don't have already in the first sample, feel free to bring them into the lab. So this is one of my favorite groups of invertebrates. And if you're thinking, it doesn't look like an invertebrate, it looks like a stick, it's because this is a caddisfly case. So there are some insects called caddisflies that make cases, and the invertebrate lives inside the case. And right now, this invertebrate is completely retracted inside its case. So often we'll see things that look like little cases made out of little tiny stones or little tiny pebbles. Some of these caddisflies even glue tiny discarded snail shells together and make these really cool spiral cases. Here's another one. Oh, and this one is poking its head out. So these are caddisflies. Not all caddisflies make cases. Some of them are free living, but many of them make cases. And this little one, the case is so thin, you can actually see him inside the rim of the case. And occasionally, like right now, he'll even poke its head out. So these make protective cases, and they live in these cases. They're called caddisflies. Caddisflies make big swarms in the spring, and some people refer to these locally as shadflies, but the term shadfly is a little bit confusing because if you go to Montreal, people there call mayflies shadflies. And when I moved to Cornwall, a lot of people were calling these things shadflies. So shadflies, depending on the geographic locality, can refer to either of these two. But both of those, whether you call shadflies these guys caddisflies, or whether you call shadflies mayflies, most people consider them annoying because of the large clouds of them in the spring. But these are two of those groups that are in that ETP, Ephemeroptera, Trichoptera, Plecoptera group. So those are the three on its own that if you get lots of these, it tells you you have reasonably healthy water. And so although they can be annoying when you're trying to go for a bike ride or sit out on the shore of the St. Lawrence River for a barbecue late in the evening, they're actually a good sign for the health of the water. All right, I'm going to see what else I have here. Got some more? Okay. A big what? Oh, okay. Yeah, so one of the things that I noticed crawling around on the outside of this caddisfly case, see if he's still there. If you noticed while I was talking about the case earlier, if you noticed something crawling up around here, it was a flatworm. Oh, I think he's right here. So this is a flatworm. Scientists call them Planaria. And Lexi just brought me one that's isolated a little bit, so we'll probably be able to see it better. So these are flatworms. And these are cool animals for a variety of reasons. So another example where common names are misleading. So they're called flatworms. True worms have segmented bodies, and I'm sure everyone can picture an earthworm with the body sections or segments. So these flatworms have no connection to earthworms. Insects are more closely related to earthworms than these Planaria. These Planaria have epidermal cells. So epidermal cells are skin cells that are ciliated. That means each cell that makes up their skin has a cilia. A cilia is like a whip-like thread, and all of these threads, all of these whip-like structures on each of their skin cells move in a coordinated fashion, and that's how it crawls along. It actually beats its cilia. Its skin is literally crawling, and that crawling skin helps it get traction and move its body along in a really smooth, graceful fashion, like you can see here. The other really interesting things about these Planaria is a closely related species, another species of planarian, is responsible for, I would say, at least half of what we understand about how molecular biology and our nervous systems work. These Planaria are actually used in medical research, and the reason they're used in medical research, both molecular genetics and understanding how nervous systems work, is they have a really simple genetic code. They have only a small number of chromosomes, and every part of those molecules has been mapped out. Some of you have probably heard about the human genome, where scientists tried to, and for the most part successfully, map the entire human genome. Well, these have such simple genomes, such simple collections of genes and chromosomes, that their genome was mapped decades before the human genome. And so they're great for doing experiments because they're complicated enough that results we get can be applied to more complex animals like humans, but they're simple enough that we know every gene, every molecule that makes up every gene, how those genes lead to expression of various body parts and behaviors of this really simple organism. So these flatworms are probably one of the most important experimental animals in the history of medicine. Oh, that's a good question. And thank you for that question about how big it is. So if you, that's it right near my thumb. And I'm a pretty big guy, so that's a fairly fat, stubby thumb, not a dainty thumb like some of you probably have. So that gives you a bit of an idea. It's a very, very tiny animal. And if I would say that the total body length is maybe a centimeter at the most. So most of these animals, and to put the others in perspective, so the largest animal I've shown you so far was this damselfly. And once again, next to my thumb, that's the end of my thumb. So even the largest animal we've shown here is pretty small. So if they look big and scary on the screen, they are not. They are all very, very tiny. All right, Lexi's bringing me something else. All right. I'm not getting a very good look at this because it's all sort of scrunched up in the ball. I think Lexi said she thought it was a small leech, but she ran out of the room before I could confirm that that's what she thought it was. So flatworms, the one I just showed you, are often confused with leeches when leeches are small. Leeches are parasites. They feed on blood. And they have a little suction disc at each end. And one end is just a suction cup for grip, and the other is connected to its mouth, and that's where its feeding apparatus and hooks are for it to dig into your skin. There are a lot of myths about leeches. Leeches don't burrow deep into the skin to feed. They latch onto the surface, they make a tiny puncture, and they extract some blood. And if you, there are also all sorts of myths about how to take leeches off, and some of them involve salt, pouring salt on it. Some of them involve burning them off. Your risk from the match or the lighter is probably greater than the risk from the leech. And oh, I got something else coming next. So if you want to get a leech off, take something flat and skinny, like a credit card or a bank card, and just scrape it along your skin and remove the leech. The reason why that's better is if you burn it or salt it, you are torturing it before you are removing it. And if you are torturing it, it's more likely to spit up its gut contents into the wound it has made, and you're more likely to get an infection. If you are fed upon by a leech, you know, like anything, some people react to animal bites and are a little bit extra sensitive. But for a typical person, when you're fed on by a leech, you just scrape it off, wash the wound if you happen to be close to a sink and a bar of soap. But other than that, leeches are pretty harmless. They don't burrow their mouths into your skin, as is commonly thought. Stephanie brought me something else. I'm not quite sure what it is she brought me. And I can't even find what dish it's in. Okay, this might be it. Ah, okay. So this is a beetle larvae. So there are lots of beetles that are aquatic even as adults. So most of these things I've shown you, except for the flatworms and the amphipods and the isopods, most of the insects like the caddisflies, the mayflies, all the insects I've shown you are aquatic as larvae or nymphs, but then they become adults when they emerge, and then they become important food for bats, which is one of the things I study, and for many of the insect-eating birds like barn swallows and tree swallows, flycatchers. Beetles can be found in the water as immature and also as adults. So this is a beetle, and I'm not going to describe why I know it's a beetle, because beetles are really diverse, and it's hard to find a rule that works to identify something as a beetle. You just need lots of experience. It's almost a process of elimination. If they're not any other kind of insect, then they're a beetle, and then eventually you learn to recognize particular species. And so beetles are the most diverse group. In fact, there are so many species of beetles on the planet that if you made a list of all the beetle species that scientists have described and a list of all non-beetle animals, so that means all the mammals, birds, fish, amphibians, reptiles, all the insects that aren't beetles, so butterflies, flies, etc., and you added those to the list with all the mammals and other vertebrates, the length of the beetle list of species would be longer. In fact, if you took only one family of beetles, the scarab beetles, and you put all the other beetles that weren't in the scarab family into the list with the birds, mammals, etc., the scarab beetle list would still be longer. So our planet is really a beetle planet, and in terms of numbers of different species. So this is a beetle larvae, and I think I might have, yes, here it is. And so this is a common beetle that we find in the river as an adult. This is called a riffle beetle. Now the adult beetle is not the same species as the larvae I showed you. So the larvae I just showed you will not turn into one of these riffle beetles, but this is a riffle beetle. In terms of size, that's not any longer than the Planaria you were looking at beside my thumb earlier. So it's a very, very tiny beetle. These are they're called riffle beetles because they like to live in kind of rapids areas where, which we call riffles. Streams alternate between fast-moving riffle areas and slower, deeper pools. And these beetles like the riffle areas. So if you find these beetles, the water is well oxygenated, and that's a good sign for fish and other things that require lots of oxygen. All right, I'm going to see what this is. So this reddish worm-like animal really is a worm. So a bloodworm that I showed you earlier is actually an insect, a type of midge. This is an aquatic worm. So this looks like an earthworm. It's an aquatic equivalent, an aquatic version of an earthworm. So the earthworms that live in our soil, they have relatives, they have related species that live in the bottom of the river. Let's see what else I've got in here. So that's an isopod I've shown you already. I'm going to go back to the amphipod. If I can find it. Oh, that's not an amphipod, but those smaller things, those really tiny things, those are various types of zooplankton. And I do not know what kind of zooplankton those are, because I rely on Stephanie for zooplankton identification, because that's one of her expertise.
Steph's gonna try and have a look and see if she can figure it out, if you want to center them under the.
Okay.
Oh.
So is she going to come in here?
She's looking at them on the screen right now.
Oh, she's looking at them on the screen. Okay.
Yeah, I think I'm gonna come in the lab just because the image will be better and it's a hard one. So I need to see the head more clearly.
All right. And while Stephanie is doing that, I'm going to put some amphipods back on and tell you some things about amphipods, which I think are particularly cool.
I think it's a Cereodaphnia.
Okay, so
it's one type of the cladoceran. So in zooplankton, we have like two big types, so cladoceran and copepods. And if anyone knows SpongeBob SquarePants, Mr. Plankton, the big green guy, that's the mean guy, he's a copepod, so he's actually a cyclopod. This one is not, oh, and we can see there's some baby in the pocket, I'll try to zoom in. So they're really cool because they actually carry their baby in their dorsal, in a dorsal pocket, and they're all clone of the mother. And so all the little things you see here where I'm pointing, those are little babies, and you actually have the little eyes here. So right now they're not moving, but when she moved, we could actually see them pretty well. And now I can see, oh, and that little guy here, that's an Ostracod. So it looks like a shell that's swimming super, super fast. And, oh, they're so weird. I don't see those often. So I'll need to check in my book quickly. I have two guests right now. Oh, and that's a little Caddisflies that just came in. And you can actually see all the digestive system, and those are the, she's filtering a lot of water right now. And I wouldn't be surprised that if this part here, that's the heartbeat. I wonder if she will move her little claw so we can see the babies. But yeah, they're really cool. I love zooplankton, if you can't tell. Oh, they're moving inside, so you can kind of see them moving a little. I don't know how good your image is on your side, but I think that's pretty neat. I can try and find like a copy of that.
So yeah, so these zooplankton that like the one that Stephanie was describing, and also the amphipods, which I showed you earlier. Now that we've got this one, I don't need to put an amphipod on, because what I wanted to say about these is these zooplankton, as well as many of the larger animals like amphipods, the females can actually reproduce by cloning themselves. They just bud off little parts of their body, and so they don't need to engage in sexual reproduction with a male. They can have babies all on their own. They do also reproduce sexually, so they can reproduce asexually by cloning, or they can reproduce sexually. And the advantage of sexual reproduction is there's always a new mixing of different combinations of genetic information. And if the progeny, the offspring produced from that sexual encounter has a combination of genes that is particularly well adapted to some new changing environment, then they win the lottery. They've got it made. The advantage of cloning, one of the advantages, if you're already the perfect biological type and form for where you live, and the environment is unchanging, then if you make clones of yourself, that female is guaranteed to have offspring that are just as well adapted as she is. So many of these invertebrates, including most zooplankton and amphipods, they kind of have the best of both worlds. They can gain the advantages of sexual reproduction when environments are changing, and they can also gain the advantages of cloning when they're already in a place to which they're perfectly adapted. So I'm going to see what else I can find in here.
I think we're gonna stop for questions now, Brian.
Okay, that's perfect, because I'm having to look harder and harder to find new things. So that's awesome. If there are questions, fire away. And do you want me to stop the sharing so that we're on full screen? Or just look at the beautiful zooplankton?
That's fine. I think, Brian, if you want to stop sharing.
Okay. All right.
Don't see any questions coming in yet.
So if there are no questions, for those who are still with us, thanks for joining us. Hope you found it interesting to see what scientists actually do on a typical day. And if you had fun, we'll be doing more of these things.
Yeah, we should be back on Friday for the second bat workshop.
Hopefully with better sound for those who are on that one. We think we've got a plan to improve our sound. Oh, so Stephanie has concluded it was a Daphne she was looking at.
All right.
Which is a type of zooplankton.
All right, and still no questions. So I think probably we'll say thank you to everybody for joining us today, and we will see you all on Friday.
Thanks, everyone. All right.